I am a pharmacist and a research scientist at the Office of Cannabis Management. We welcome you to today's multidisciplinary research driven webinar series called Rooted in Research, where we discuss the latest advancements in cannabis research, such as emerging trends, evidence based findings, challenges in cannabis research, and regulatory barriers. But before we start, I will read a disclaimer regarding the webinar. Um, the views and opinions expressed in this live event are those of speakers and do not necessarily reflect the views or positions of the New York State Office of Cannabis Management. The New York State Office of Cannabis Management makes no claims, promises, or guarantees about the accuracy, completeness, or adequacy of the contents and expressly disclaims liability of the opinions expressed. The appearance of these speakers is for the information and convenience of the public and does not constitute endorsement, recommendation, or favoring by the New York State Office of Cannabis Management. So today, our presenter is the very accomplished Dr. Daniela Vergara, and she is an evolutionary biologist, data analyst, educator, scientific writer, and public speaker, and serves as an international consultant, policy advisor, and expert witness for cannabis related legal cases in the US and Latin America. In addition to her multiple publications, she serves on the board of the 501c3 nonprofit Agricultural Genomics Foundation, AGF, which is dedicated to making cannabis sativa science accessible to the public. Through AGF, Dr. Vergara engages in public science education and currently leads a national collaborative cultivation project in partnership with Atlas Seed. She is fluent in Spanish and English, and she actively translates her work for broader Hispanic audiences. Her research and outreach are made possible through the support of private donors to AGF. She is currently a lecturer and a researcher at the University of Colorado and co-teaches basic cannabis science and advanced cannabis science courses at CU Boulder, and previously taught cannabis comparative genomics in 2021. In late 2021, Dr. Vergara joined Cornell University's Harvest New York team as a hemp specialist, where she currently supports farmers with hemp production. She has led the development of a comprehensive cannabis sativa manual in both Spanish and English, which has been downloaded more than 6,000 times, as well as a separate guidebook for hemp farmers. She is now working on an updated edition of the manual to incorporate the latest research in cannabis sativa. Dr. Vergara has also been a part of scientific teams at leading private companies, including Steep Hill Incorporated, a global leader in agricultural testing, and a biotechnology firm, Front Range Biosciences. Her research has been featured in major media outlets, including The Atlantic, Science, and 538. Most recently, she co-authored a review on sex determination and expression in cannabis sativa with a colleague from Cornell University. So with that, I am so excited to turn this over to Dr. Vergara for her exciting presentation titled Evolution for Everyone, How Natural Selection, Genetic Drift, and the Grower Participation Can Shape Cannabis Research. Thank you, Dr. Vergara.
Hi, everyone. Thank you very much for that wonderful introduction. I hope that you all can see my screen. Is my screen there? Yeah. Hi, Tom. Thank you very much for being there. It's really nice that you are around. So supportive. Okay, so evolution for everyone, how natural selection, genetic drift, and grower participation shape cannabis research. And I'm going to start with this slide that if you studied evolutionary biology like me, it may be a little bit cliche, but I hope that I put this together throughout my talk. And this is not a quote, but it was an essay that was written by Dobzhansky in 1973. Nothing in biology makes sense except in the light of evolution. Yes, I hope that throughout my talk, this quote becomes relevant. And also, I continue with this slide, right? Phenotype, any phenotype. So any phenotype is a product of genes and environment. Phenotypes are the traits that we can measure, right? So height, we can measure with a meter stick, or we can measure weight with a scale, or we can measure THC or CBD with an HPLC or with a GC. All of those are phenotypes. And the phenotype, right, that trait, that physical characteristic, is a product of the genetic makeup, right? So nature and the environment nurture, right? Genes and environment. So cannabis sativa is an angiosperm plant. So those are the ones that produce flowers. It's part of the family Cannabiaceae along with humulus. Humulus lupulus is a really important ingredient for beer. They share an ancestor that lived approximately 25 million years. There's some new estimates that suggest 27 or 28 million years. These two share an ancestor with another genus from the family Cannabiaceae, Trema. Approximately 33 million years ago was the split, and then with Celtis, approximately 37 million years ago. So to make sure that we're all on the same page, cannabis sativa is part of the family Cannabiaceae and the order Rosales, and it's a plant, right? And something that I find really interesting about cannabis is that it has autoflowers and photoperiods, and probably you are growing either one or both of these, right? The autoflowers are those that do not depend on the light cues to flower. The speculation is that it has genes from ruderalis, that it's supposed to be from Siberia, and when you have plants in those very northern latitudes, you either have a lot of sun or very little sun, so you should not depend on the light cues to flower. The autoflowers do not require as much trellising and pruning, so as much hand labor, and they usually grow smaller than the photoperiods that do depend on the light cues to flower. Again, these ones grow much bigger than the autoflowers. If you start them, for example, around this time and harvest in October, you are going to have probably big Christmas tree if you're cultivating on the ground, right? So again, the autoflowers, because they're much smaller, they don't require as much hand labor. And this is a picture, some autoflowers and some photoperiods here from New York State. So my aims for this talk is to tell you about some of the questions that I have that I think are relevant and interesting. To talk about how can we all participate in collective research and to address some misconceptions and misnaming. As Sam was saying, I am Dr. Daniela Vergara. I am part of the Cornell Cooperative Extension, the Harvest New York team. I am the hemp specialist. This is my emails, my website, which is a little bit outdated, but you can find who I am there in that website, my social media, Instagram and LinkedIn. And so all of my journey in cannabis started at the University of Colorado in Boulder back in 2013, where I started as a postdoc. There, we founded the Cannabis Genomic Research Initiative. I'm a co-founder, which is a group that we try to understand the cannabis genome. I am a board member of the Agricultural Genomics Foundation, which is a 501c3 non-for-profit organization that aims in bringing science to a broad public. And I am also part, as I was saying, with Cornell Cooperative Extension, the Harvest New York team. I am the hemp specialist. And I was talking about the autoflowers and the photoperiods, right? And now I'm going to talk about what are the questions that I have that I find interesting. And this is an actual question that we posed for a grant that unfortunately we didn't get, but I still think that this question is very relevant. And the questions were, what is the production per farm? What is the production in New York State or in the US or in the world? How much does each particular plant yield, maybe in weight and in cannabinoid content? And as you all know, you can have a plant that is very big but has very little THC, or a plant that is smaller but has a lot of THC, right? So how do we estimate yield? And could autoflowers yield more than photoperiods per unit area? And maybe could they be more profitable? Again, because perhaps they require less hand labor, less trellising or pruning, etc. So those were questions that we had and that we posed through Cornell. Again, unfortunately, we didn't get the grant, but I'm going to tell you about what was it about. So we wanted to know the yield and profitability of autoflowers and photo periods. So we wanted to have 2 plantings of autoflowers and 1 of photo periods in high tunnels. And so these high tunnels these structures are kind of a greenhouse, but you grow on the ground, right? So we were going to have a randomized block, and we had different spaces for the autoflowers and the photo periods. We were going to have a total of 160 plants per year. We were going to have 2 high tunnels. And we had the autoflowers, the first set planted in early June, maybe late May. And then the second one, maybe in early August. All of the photo periods were going to be planted in early June. And the last set of autoflowers and the photo periods were going to be harvested in around October, right? So we were going to have different spacing between the plants. The autoflowers at 2 feet and at 3 feet, right? And here you can see the diagram of the tunnel, right? So we were going to have the autoflowers at 2 feet and at 3 feet, and then the photo periods at 4 feet and 6 feet, right? The idea was to measure yield, right? Whether the plants that were grown at 6 feet, for example, were bigger than those at 4 feet, right? That's what we expected, and we were going to test for that. So we were going to assess yield and height and stem diameter and weight, dry weight, wet weight and dry weight, and bucked weight and waste weight. And then cannabinoid content, we have a purple probe, but we were going to do full lab profile on 24 representative samples. And then we were going to do foliar analysis to see whether the plants were taking nutrients. We were going to have cover crops and see whether these cover crops helped with the nutrients and with the soil. And we were going to measure the pH of the soil, and everything was going to be analyzed through R, which is the program that I use for analysis, via two-way ANOVA. And so these questions, right, so are questions that I, again, think are very relevant, and leads me to the next part. How can we all participate in collective research, right? Because those questions that we were asking through Cornell are questions that I think are very relevant for you, and perhaps at some point this could be done with farmers, right? With real farmers having these questions. So now, how can you be part of research? So right now, we have a reciprocal transplant experiment through the Agricultural Genomics Foundation. And I'm going to tell you the overall goals of these reciprocal transplant experiments. So the main goal in general of these experiments is to understand whether populations or species are adapted to a particular environment. So what we do in this type of experiment is to observe the performance in different environments and determine if these differences in performance are due to genes or to the environment, right? So we have different strains that are grown in different environments. So strain number 1, strain number 2, strain number N, however many, are grown in environment 1, in environment 2, and in environment N, right? And then you measure whatever it is that you're going to measure in the phenotype. Again, you measure the phenotype, and we figure out whether one strain does better than the other one or not. So for this reciprocal transplant experiment, we were going to study how environments affect cannabis sativa, assess yield, quality, disease susceptibility or resistance, cannabinoid and terpene profiles, and we are going to use statistical analysis to guide these breeding improvements, ideally, right? We are partnering with Atlas Seeds. They are our seed provider. Ideally, the plants would be grown throughout a protocol that we are providing. We would collect data on germination, growth, disease, different harvest metrics. the growers would share environmental conditions and cultivation practices, and comply with state, federal laws, and agree to possible site visits if it's possible, right? The requirements to participate in this experiment that is right now, it's ongoing, right? So you can sign up for this experiment. So the requirements is to purchase these seeds from Atlas Seeds at a discount. Thankfully, they are giving us a discount for these experiments. You need to have basic cultivation tools, and I'm assuming that if someone is buying these seeds from Atlas Seeds, they already have cultivation tools. We have different varieties that are autoflowers, quicks, and photo periods. We would give data. We would give a spreadsheet to collect the data. And so what is required is that you have online tools to participate, that you have internet, that you know how to use some Excel or Google Sheets. And ideally to share results and cultivation details. For example, if you have soil tests or chemotypes tests. So for those people that are in New York, this is data that you already report to the OCM, right? How much did you produce, the weights, how many plants did you plant from this particular strain? What was your waste weight, your bucked weight, etc., right? Ideally, we would also know, you would also tell us the A and B and C bud distribution. And there's optional data that would be very helpful. For example, sale data or prices, right? How much did you sell them, or did it go for biomass extraction? So with this type of experiment, we're trying to support farmer-led research and community-owned data, right? AGF, the Agricultural Genomics Foundation, is a 501c3 nonprofit. So we're trying to do this as a community, as a collective. We're trying to create open and inclusive industry pathways and to contribute to educational materials and opportunity for the industry. What AGF, the non-for-profit, would provide are the instructions and the support throughout the study. We would be analyzing the data and sharing the findings. We would be recommending based on the results, and all of the data would be securely stored. All of the farm identities remain confidential. And something that I forgot to say is that AGF does have a research license with the OCM, and we're doing that through that research license, right? So going back to the goals, the general goals of a reciprocal transplant experiment, right? We're trying to understand adaptation, to observe the performances in different environments, and to determine if these differences are due to genes or the environment, right? And in this case, we have 10 different strains. So here we don't have strain N, we have 10 different strains because we chose, along with Atlas Seeds, we chose those 10 different strains, right? And the environment N depends on the number of farmers that we are getting, right? So environment N is N equals the number of farmers that we get. And we would observe the performance in these different environments. For example, are some strains better at particular location? Do we find that there's differences in disease pressure? Do we find that maybe someone that grew indoors have different diseases that someone that grew outdoors in the different strains, right? Those would be very interesting questions. And so with this experiment, we would understand, for example, yield across these varieties and environments. We could measure the production per plant, ideally, and per farm for the different types, for the autoflowers, the quicks, and the photos. We could compare total yield across sites to estimate production per acre or per farm, and assess the yield per unit area for quicks and photos and autos. We could analyze, if we get this data, right, from THC and CBD and cannabinoid profiles, and correlate them with the environmental conditions. And the environmental conditions in this case are the different cultivation practices and the different locations, right? We could determine whether the cannabinoid content differs between the varieties and the growing conditions. And we could know, for example, if the A and B and C buds are produced by each of the plant, or by variety, or by location, and see the commercial viability per different strain. We could potentially, depending on the data that we get, we could potentially evaluate labor and economic efficiency, so the labor inputs and the cultivation practices. Again, because usually autoflowers do not need trellising or pruning, we could add those costs and estimate those costs per gram. We could test whether the different types be more profitable because of labor, maybe shorter cycles, right? Maybe with the autoflowers, you can have 2 cycles as the experiment that I talked about, the beginning, the experiment that we proposed, right? Could you have 2 cycles of autoflowers? Maybe you could have even 2 cycles of quicks. I don't know, right? And these are the questions that we're trying to answer with this transplant experiment. And again, we could gather sales and pricing information to estimate revenue. And there is some information that is required to participate, but the sales and the pricing information is optional. I understand that this is a little bit tricky for some people, so that some of the information we would love to have, but it's optional. So ideally, we would be able to understand local data and regional context, right? What is going on in New York State, and especially because New York State has these different microclimates, so that would be very interesting to understand. And we can use this data to understand the production potential per farm, and the estimates for production. And again, to develop farmer driven recommendations. At the end of the day, it's you, the farmers, that are growing these plants and that will be giving us that data. So this is farmer driven 100%, and it's based on real world data, right? This is data that you guys are giving us, that at the end of the day you are selling, right? So this is real world data, and this would contribute to community owned data to inform sustainable production practices, ideally, right? And to provide science based guidance. Perhaps we are able to understand whether one strain is better than another one in a particular location. And that would help us with profitability, right? And sustainability. And ideally, for choosing resilient varieties by location. So if you want to participate in this experiment, you can sign up here. This QR code would take you to the website. There was a glitch in the website, so if it doesn't work, please email us. Anna is another board member from AGF. So this is Anna's email and this is my email. So please help us spread the word and sign up. And again, what we're doing with this experiment is understanding the phenotype, because again, the phenotype is a product of genes and environment. And if we're assuming that these varieties, which have been bred by a very reputable company, that these varieties, per variety, you have the same genes, right? Then what is changing is the environment that is provided by the different farms. So we're looking at how this phenotype is going to change depending on the environment. So that is a very powerful way to understand how these different varieties behave, right? That's why it's called a reciprocal transplant experiment. We're taking these different varieties and transplanting them in different locations. And I'm going to show you here, this is what we could have a possible result. This is called a norm of reaction in evolutionary biology. And what we see here in the x-axis, which is the horizontal axis, are the different environments. And in the y-axis, the vertical axis, we have any phenotypic trait, let's say height or weight or whatever it is, right? THC content. And so you can see, for example, that strain number 2 in environment 1 it did really poorly, but in environment N, right, in that farm from farmer N, it did really good. Strain 1, on the other hand, in environment 1 did really good, but did really bad in environment N, right? This is something that we could do with all of these different traits, okay? And that leads me to talk about evolutionary biology. And this is the last part of my talk to address misconceptions and misnamings. I don't know, I'm gonna, because I can see only my screen, I'm gonna see whether there's anything in the chat. Oh my God, there's so many things in the chat. Wow. I am sure those. Okay, I am going to address the chat at the end, but anyway, okay. So now I'm going to talk about evolutionary biology. So what is evolution? Evolution is change through time. And it is the change of heritable characteristics of populations over generations. So that sentence there is very important because one, it is heritable, right? So it's those changes, those traits that are inherited from parents to offspring. Now, in populations, and notice that populations is bolded because this doesn't happen at an individual level. It happens at a population level, okay? So it's not one individual, it's a population. And then over generations. So the time is measured in generations, okay? So if we're talking about sharks, the generation time is much longer than that of bacteria. So if you do experimental evolution in the lab, you do not work with sharks. You do not work with whales or turtles or oak trees. You work with bacteria or you work with nematodes, where the generation time is 2 hours, right? In the figure here, we have Darwin's finches from the Galapagos Island, that they evolved from one common ancestor that lived approximately 1 to 5 million years ago. So in other words, all of these populations of finches arose from this one single population that lived around 1 to 5 million years ago. And all of these populations evolved. So all of these populations changed through time based on selection of their food, right? So that's why they have different beaks. And the beaks are adapted to a particular food. So they specialize on eating different things, and therefore their beaks are different. Okay. So again, when you think about evolution, think about heritable, think about population, and think about generations. Okay. Time in generations, a big group of population, and the changes are heritable. The theory of evolution was independently conceived by two different people, Charles Darwin and Alfred Wallace. However, Darwin has received most of the credit because in 1859 he published this book on the origin of species. Wallace had some misfortune. I think that his boat, the boat where he had most of his data, sank, or it got fire, or something happened to that boat, or maybe it got fire and then sank. But in any case, we know Darwin, but we really do not know Wallace, although Darwin did find about the work of Wallace, and that's when he published his book like ASAP. But in any case, these two people conceived independently the theory of evolution. Natural selection. Natural selection is the differential survival and reproduction of individuals due to differences in phenotype. It is a mechanism that drives the evolutionary process. It's a mechanism that drives the change. So the organisms that are more adapted to their environment are more likely to survive and to pass the genes, right? I'm going to give two examples of natural selection. These are the examples that I give to my class that I teach online. So here you have a fox that eats mice. So the fox eats the mice, and then the ground, notice that the ground is very dark. So the fox sees the white mice more than the dark mice. So when you start with your mice population, right, you have dark mice and white mice. But since the fox is better at catching the white mice, right, so the white mice are selected against, then the population of dark mice increases in frequency, okay? And so we're assuming again that the color of the mice is heritable, so that the dark mice are going to have dark baby mice, okay? Now, in this other example, we have a tree and we have giraffes, and those giraffes that have long necks can reach the top of the tree, which has more nutritious or more food, right? So assuming that the length of the neck is a heritable trait, and so that those giraffes that have long necks are going to have babies that have long necks, then this trait is going to increase in the population, okay? Natural selection, again, is a mechanism for evolutionary change. It is directed, it can be predicted and calculated. You can estimate heritability with a number, with math, okay? And it is stronger in larger populations. The requirements for evolution via natural selection, so the requirements for change via natural selection, is that you must have variation in your population, right? The beaks need to be different, the length of the neck needs to be different, that the variation is heritable from parents to offspring, and that it has an effect on fitness, right? That there's some sort of competition where those individuals that are better adapted have babies or have more babies, right? So there's more variation. So individuals in the population have different phenotypes, inheritance. The offspring inherit these traits from their parents, and then there's some sort of competition. These are the conditions for natural selection. Because again, right, we have all of these different phenotypes that are inherited, the genes, in a particular environment. And now I'm going back to our reciprocal transplant experiment. This is one of the reasons why I think it's really interesting, because we're in understanding adaptation. We're understanding whether some individuals are better at a particular environment, right? We're going to observe the performance, the yield in these different environments, and determine if there are some genes that are better in a particular environment than in another environment. Because again, nothing in biology makes sense except in the light of evolution. Okay? Now let's talk about artificial selection. Artificial selection is when humans, so when we perform the selection, right? We decide who reproduces depending on what we want, right? And we have all of these different breeds of cats, for example. We also have we have artificial selection in corn and in strawberries and in seedless watermelons, which is mind-blowing in my opinion. And it seems like we have some sort of selection in THC, for example. So this is a graph that I modified from these two papers that are not open source. So I had to modify it so I can show it to the public. But Volkov et al. 2014 and El Solhi 2021 show the same thing. We have time, again, on the X, the horizontal axis, and on the Y-axis, we have THC percent, and we can see that THC increases through time. So breeders and growers, whether knowingly or not, have selected those plants that have the most THC for them to be the parents of next generation's offspring. So again, here we are assuming that the trait, the THC, is a heritable trait, okay? So a comparison between natural selection and artificial selection. So natural selection occurs without human interference, right? It's the fox or it's the tree that has more nutritious food in the top. And the environment is the one that selects, that chooses the individuals that reproduce or that reproduce more. Artificial selection is humans that select those individuals that reproduce depending on the traits that we want, right? But in both cases, in both natural selection and artificial selection, the parental traits are inherited by the offspring, right? There is heritability. This results in differences in a population, again, a population level, and this genetic variation yields to phenotypic variation. And time is measured in generations. Again, we've performed artificial selection in dogs, and this is my favorite example, Brassica, which is a wild mustard, and we selected for the flower, and we have cauliflower. We selected for the lateral buds, and we have Brussels sprouts. We selected for the leaves, and we have kale. But at the end of the day, is the same wild mustard, because again, we're choosing based on the phenotype. We're choosing those genes for a particular environment. Again, here we go, a reciprocal transplant experiment. And for New York, for example, ideally, we would be able to understand disease, which varieties are better or not better for a particular environment based on disease resistance. Perhaps you already have a favorite one that does not get powdery mildew or gray mold or septoria, which is a big one. Here's Julian Smith, who has grown a beautiful field near Buffalo, New York. And we can see his beautiful field. But in New York, we have not done this selection in cannabis, right? And New York, again, has these microclimates, okay? So now this is going to take me to talk about genetic drift. And I'm talking about genetic drift because, as an evolutionary biologist, is my duty to explain what genetic drift is, okay? We were talking about the mechanisms of evolutionary change, and that natural selection is one of those mechanisms for change to happen. Well, genetic drift is another one of those changes. It's more complicated, and genetic drift is actually called random genetic drift. And before I explain genetic drift, I need to explain what an allele is. So an allele is a form of a gene, right? And the gene is found in a particular position that it's called a locus. So let's assume that we have this particular position for, I don't know, let's say hair, the gene for hair, and we inherit a form of a gene, an allele, from mom and another one from dad. In this case, one of the alleles is red and the other one is burgundy, okay? We can have multiple different alleles, and this is usually how we think about them, that we have homozygous, big A, or we have heterozygous, big A, little a. So in this case, big A, both mom and dad gave us the same form, the same allele. In this other case, one of our parents gave us a big A, the other parent gave us a little a. And in this last case, when we're homozygous for the other allele, for little a, so again, both parents gave us the same form of the gene, little a and little a. This was brought to us by Mendel, who was a monk who crossed peas. And so interestingly, one of the reasons why Dobzhansky is really important is because he was part of something called the modern synthesis, which took Mendel's ideas and Darwin's ideas and put them together at some point in the, 1940s, around there. So that's really interesting because Darwin did not know about Mendel. But in any case, Mendel crossed peas. So he crossed, for example, this purple pea with this white pea, and all of the first generation were purple, right? So if we see it in this Punnett square, we have that everyone is PW, allele P, and allele W, right? But then when he crossed this first generation, he gets that 1/4 of the individuals are white, right? So then if we look at the Punnett square, we have that that 1/4 of the population is big P, right? Homozygous. The other 1/4 of the population, the other 1/2 of the population, is heterozygous, so PW, and then this last 1/4 is WW, okay? Now that we know what an allele is, that alleles are forms of genes, right? Let's talk about random genetic drift. So, mechanisms for evolutionary change: selection and drift. Selection is directed, can be predicted and calculated, and it's stronger in larger populations. Drift is random, and it's stronger in smaller populations. So what is drift? Drift is an evolutionary process that changes the frequency of existing alleles in a population due to the random sampling of organisms. So that is very loaded, but it is basically a bottleneck effect. So just imagine that you have this bottle that is full with different alleles, right? And you have allele blue, allele yellow, allele red, and by chance, right? Just by chance, because there's a bottleneck effect, you sample these alleles from this population, and you're sampling them, and then at the end you end up with a new population that does not have the red allele. The red allele is lost, and that's why drift is stronger in smaller populations. So again, another example with a bottle, you have your original population, you have this bottleneck event where you lose, in this case, allele green, right? And you can see it in this graph. You have time, again, on the x-axis, the horizontal axis. In the y-axis, you have population size, and there's this bottleneck event, so your population shrinks, and perhaps you can lose entirely that allele. So it's extinct. Perhaps your population recovers and you don't lose all of the alleles, right? So when you lose the allele, it's extinct. When everyone in your population has that allele, it's called fixation, right? So in this case, it seems that the blue allele is going to reach fixation. I think about genetic drift through this Punnett square, and I'm imagining the smallest population have a population size that you can have, which is a population size of 2. And in this case, we have one parent that has alleles A and B. The other parent also has alleles A and B. So we have that 1/4 of your population is AA, 1/2 is AB, and then the other quarter is BB. By random chance, you lose BB. It died. A log fell on it and it died. Then the next generation, you have AA reproducing with AB, right? So half of your population is going to be AA and half of it is going to be AB. But allele B could potentially be lost in your population, okay? So when people say, and this happens often to me when I go to a grow and they say that individual was a victim of genetic drift, why is this a wrong statement? Again, hint, what is genetic drift? It is not a weapon. Drift is not a weapon. Drift is a mechanism that acts at a population level. And because it is random, the alleles that would be lost or fixed cannot be predicted. Since drift acts on a population level by observing the phenotype of one single individual, we cannot conclude whether the alleles are lost or fixed or basically anything, right? Marijuana crops that are grown by seeds, that are not grown by seeds, that are grown by cloning, by cuttings, do not have the opportunity to recombine, and therefore, if there's no recombination, there's no drift, right? So people usually confuse drift with the possibility of accumulating deleterious mutations that may happen through cloning. So when you are referring to drift, you're probably referring to mutation accumulation. And we do have at least 3 papers, to my knowledge, that talk about mutation accumulation in cloning when you do cuttings, right? So cloning over multiple generations may cause deleterious mutations to accumulate. Regarding drift, we do have some empirical examples of drift, right, in snails. E. coli, which is a bacteria, Drosophila melanogaster, which is the fruit fly, the mitochondrial genome, C. elegans, which is a nematode. So we do have those examples. And now that I gave you my lecture on evolutionary biology, again, here's my contact information, my emails, my social media, LinkedIn. And with that, I am going to look at the chat. Thank you.
Okay, great. Thank you, Dr. Vergara. That was an amazing presentation, and it really brought me back to my days that I was taking biology. So thank you for that. So I think at this time, we'd like to open it up to the question answer portion of the webinar. So we do have a question here. So are there still wild native populations of cannabis sativa? If so, where are they?
We don't know whether there are wild native populations of Cannabis sativa. We don't know if there are.
Okay. Our next question is, how does understanding mechanisms like natural selection or genetic drift help us interpret modern cannabis cultivation and breeding practices?
Oh, this is great. Well, I guess that I did talk about this a little bit, about how selection, how can we use selection for breeding, right? Choosing those individuals that are best to our environment. So I think that that is important to understand. Now, about drift, I was talking about drift because I believe that that's, at least in my experience, that's um wording that people use without really knowing what it means. And I take it on me, I'm an educator and I am an evolutionary biologist, and it should be me, the one that clarifies that genetic drift is not mutation accumulation. So that's why I took this opportunity. Thanks OCM for allowing me to talk about genetic drift. I Yeah, so yeah, that's the reason why I was talking about drift, is because I hear it all the time and people do not really understand what it means.
Yeah. Okay. You emphasize that phenotype is a result of genes interacting with the environment. How can growers use this knowledge to improve yields or plant quality?
Well, so you can modify the environment or you can breed for particular plants in your environment, right? So if you um I guess that if you have an indoor grow, you have more or less a constant environment, and you then can choose the varieties that do the best on your indoor grow. If you are growing outdoors, you can still choose the varieties that do best. For example, I do think that the varieties that were bred in California or in Colorado may not be the best here in New York. I do think that breeding needs to happen here in New York for New York climates, especially because New York has these different microclimates, right? So I do think that that understanding what varieties are you using and your environment is crucial in order to improve your yield and whatever it is that you want to improve, right?
Okay. And are there particular environmental factors you found to be especially influential in cannabinoid or terpene expression?
So there was a paper in 2023, Sankarimi et al., that found that plants that were grown outdoors produced more terps than the ones grown indoors. So there you go. Can
Okay,
I
great.
can I look at the comments in the chat? And then, okay, thank you, Charmaine. Thank you. Hardcore Haze, why wouldn't she mention that Humulus lupus is called hops? I'm sorry, my mistake. Lupulus is hops. Hardcore, very good question. Yes, veggies out front. That is a really good question, actually. Um, eh, okay. If I was a wheat grower, I would love some Cornell level research. Here you go. Here you have me. What field of, okay, I think that those were new comments. Okay. I think that those, I don't know if there were more comments, but those were the ones that I read. Thank you.
Okay. Well, again, we thank you very much, Dr. Vergara. So I just want to remind everyone as we conclude that today's replay will be available right after on our YouTube channel, LinkedIn, and Facebook. We also want you to stay connected to us by signing up for our newsletter. And we will also be putting up a speaker interest form. So anyone who is interested in presenting their research, please fill out this form or forward it to your colleagues or friends that are doing research in cannabis. We will also be sending out a post-webinar survey, which will be emailed to you. So please fill it out. Take a few moments. Please fill it out so we could take your feedback and improve our webinars. Or we want to know what you guys want to hear about. And for the month of June, please stay tuned for our next Rooted in Research, and you will find that information on our socials as well. So stay tuned for that information. So once again, thank you very much, everyone, for joining us today for this wonderful webinar. And we'll see you next time. Thank you so much.